Hello students. Today you have the last lesson, the last topic in this semester, and the name of it is food poisoning. Food poisoning is illness caused by the infection with microorganisms and ingestion of toxins produced and chemical poisoning. You should know that the causes of food poisoning are bacteria and their toxins, viruses, chemicals, and vegetable poisoning. Food poisoning, or another we can say foodborne illness, foods contaminated with pathogenic microorganisms usually do not look bad, taste bad, or smell bad. It's impossible to determine whether a food is contaminated with pathogenic microorganisms without microbiological testing. You should know that classification of foodborne diseases. They are classified into two types: foodborne infections and foodborne intoxications. And the difference between these two classification, foodborne infection is caused by the ingestion of food containing live bacteria, which grow and establish themselves in the human intestinal tract. From other side, foodborne intoxication is caused by ingesting food containing toxins formed by bacteria, which resulted from the bacterial growth in the food item. The live microorganism doesn't have to be consumed. Now, more deeply. So, foodborne infection. These can either be bacterial, viral, parasitic. Foodborne infections tend to have long incubation periods and are usually characterized by fever. Bacterial foodborne infections. Bacteria are the most troublesome and important biological foodborne hazards for the food service and food retail establishment. Bacterial cells can exist in two different states: the vegetative state and the spore state. Spores are produced when the bacterial cell is in an environment where it cannot grow, like frozen foods or dry foods. And the first example is Escherichia coli. When we study about water supply, we study about Escherichia coli, coli titer, coli index. But also it can be a foodborne disease. It can be the cause of foodborne disease. The Escherichia coli group of bacteria includes 4 strains of foodborne pathogens: enterotoxigenic, enteropathogenic, enterohemorrhagic, and enteroinvasive Escherichia coli. And the most important of the group is a particular type of enterohemorrhagic Escherichia coli called E. coli O157H7. Illness can be due to an infection and a toxic infection. Illness due to Escherichia coli of this strain is particularly serious in infants because it can cause kidney failure and bloody diarrhea. The common foods are raw milk and raw ground beef. How they transmit? Escherichia coli is usually transferred to foods like beef by contact with the intestines of animals. Transmission can also occur if employees are carriers and do not wash their hands properly after going to the bathroom. How to prevent? Undercooked or raw hamburger, ground beef, has been implicated in many of the documented outbreaks. However, Escherichia coli O157H7 outbreaks have implicated alfalfa sprouts, unpasteurized fruit juices, dry-cured salami, lettuce, game meat, and cheese curds. Also raw milk. So to prevent, you make sure that employees practice good personal hygiene. So for the first, clean, separate, cook, and chill. Next one, Salmonella species. Salmonella is a facultative anaerobic bacterium that frequently causes a foodborne infection. Like Escherichia coli is a source for Salmonella is the intestinal tracts of warm-blooded animals. Common foods. This microorganism exists in many foods, especially raw meat and poultry products, eggs, milk, dairy products, pork, milk chocolate, and cream-filled desserts. Before you use eggs, try to clean it, because eggs is the most spread common food where you can find Salmonella. Transmission to foods is very common by cross-contamination from raw foods, especially poultry, from food contact surfaces, cutting boards, or from food handlers. So you should know how to prevent. Cook foods thoroughly, practice good personal hygiene, clean and sanitize food contact surfaces after use with raw foods, and clean eggs before using. Next one, Shigella species. Shigella is a facultative anaerobic bacterium that causes about 10% of foodborne illnesses in the US. The microorganism is frequently found in the intestines of humans and warm-blooded animals. The microorganisms can cause an infection or toxic infection. A common illness caused by Shigella is bacillary dysentery. Where we can find this microorganism is common in ready-to-eat salads, for example, potato, chicken, milk in dairy products, poultry, and raw vegetables. What about transmission? Water that is contaminated by fecal material and unsanitary handling by food workers are common transmission routes. You should know how to prevent. For the first, practice good personal hygiene and wash foods with potable water supply, suitable for drinking. Go on. Campylobacter jejuni. Campylobacter jejuni is considered by many food scientists as the #1 agent that causes foodborne illness. The microorganism which causes infection is unique compared to most other foodborne pathogens because it has a very strict gaseous requirement for growth. It's classified as a microaerophile because it can tolerate only 3–6% oxygen for growth. This microorganism is commonly found in raw milk and in raw chicken. Some scientists estimate that Campylobacter jejuni may be present in nearly 100% of retail chickens. What about transmission? It is often transferred from raw meats to other foods by cross-contamination. This is typically done by transfer from a food contact surface, such as a cutting board, or from food workers' hands. Campylobacteriosis is the name of the illness caused by this bacteria. It's also often known as Campylobacter enteritis or gastroenteritis. This bacterial infection causes diarrhea, which may be watery or sticky, and can contain blood and fecal leukocytes. Other symptoms often present are fever, abdominal pain, nausea, headache, and muscle pain. How to prevent? Cook raw meats properly. Don't use raw unpasteurized milk. Thoroughly clean food contact surfaces, cutting boards, and hands after handling raw foods. Next, Bacterial Listeria monocytogenes. This is a facultative anaerobic bacterium that causes foodborne infection. And the name of this illness, listeriosis, that caused by Listeria monocytogenes, usually causes gastrointestinal symptoms for the healthy adult. However, disease complications can be life-threatening: septicemia, meningitis, encephalitis for people with weakened immune systems. It's important to food retail operations because it can survive under many conditions, such as high-salt foods. Unlike many other foodborne pathogens, it can grow at refrigerated temperatures below 5 degrees. This microorganism has been isolated from many foods and is most common in raw meats, raw poultry, dairy products, cheeses, ice cream, raw milk, raw vegetables, and seafood. What about transmission? Transmission to foods can occur by cross-contamination, and also foods that are not cooked properly can contain live cells. How to prevent? Cook food thoroughly and practice good personal hygiene. Vibrio species. There are 3 species within the Vibrio group of bacteria that cause been implicated in foodborne infections. They include Vibrio cholera, Vibrio parahemolyticus, and Vibrio vulnificus. All are important since they are very resistant to salt and are common in seafood. Vibrio species are commonly found in raw, under-processed, improperly handled, contaminated fish and shellfish. These bacteria are generally found more in the summer months and from warmer waters. Since the microorganism exists in many raw seafood, transmission to other foods by cross-contamination is a concern. Most illnesses are caused due to eating raw or undercooked seafood, especially oysters. Prevention. Cook seafood properly. Avoid consumption of raw seafood. Practice good personal hygiene. What about symptoms? Diarrhea, abdominal cramps, nausea, vomiting, headache, fever, and chills may be associated with infections caused by this organism. The illness is usually mild or moderate, although some cases may require hospitalization. Next, classification of foodborne infections is viral. Foodborne viruses differ from foodborne bacteria. They are the smallest and the simplest form of life known. Unlike bacteria, they don't reproduce or grow in foods. They are usually transferred from one food to another, from a food handler to a food, or from a water supply to a food. There are 5 types of hepatitis, and hepatitis A is a foodborne virus that is associated with many foodborne infections. It's a particularly important hazard to retail food establishment because it has an incubation period of 10-45 days. This means that a food worker can harbor the microorganism for up to 6 weeks and not show symptoms of illness. However, during this time, the food worker can contaminate foods and other workers in the food retail establishment. Ready-to-eat foods that are washed with a non-potable water supply or foods that are handled excessively can be contaminated with hepatitis A. Examples include raw vegetables and raw seafood. Due to the long incubation period, it's very difficult to identify the food source of a hepatitis A infection. But how long does acute hepatitis last? Less than 6 months. Transmission. The virus is primarily transmitted from person-to-person contact by cross-contamination and by fecal contamination. How to prevent? Handle and cook foods properly, avoid consumption of raw seafood, practice good personal hygiene. Next one, Norwalk virus. The Norwalk virus is other common foodborne virus that associated with many foodborne infections, with some outbreaks involving up to 3,000 people. The common foods is raw seafood, raw fruits and vegetables that are washed with a contaminated water supply, non-heated foods that are handled by people who are shedding the virus. Transmission. People who are infected can be spread directly to other people or can contaminate food or drinks they prepare for other people. How to prevent? Handle and cook foods properly, avoid consumption of raw seafood, practice good personal hygiene. Next one from this classification, foodborne parasites. Parasitic infection is far less common than bacterial or viral foodborne illness. There are many examples of parasites that can enter the food system and cause foodborne illness. Giardia. This microorganism is important because it's common cause of non-bacterial diarrhea in the United States of America. These parasites are most commonly associated with the consumption of contaminated water. Raw foods that are in contact with contaminated water, especially raw vegetables, can also be contaminated. These parasites are transmitted from a contaminated water supply, person-to-person contact, and by fecal contamination. How to prevent, handle, and cook foods properly. Toxoplasma gondii is the third leading cause of death from foodborne disease. The infective stage, oocyst, develops in the gut of the cat. The oocysts are then shed into the environment with cat feces. How do people get toxoplasmosis? By consuming foods such as raw or undercooked meats, especially pork, lamb, or wild game. The time that symptoms appear varies, but generally symptoms will appear 1 week to 1 month after consuming the parasite. You should know how to prevent. Wear clean latex gloves when handling raw meats. Cook all raw beef, pork, lamb, and veal steaks to a minimum internal temperature of 63 degrees. Wash hands thoroughly with warm water and soap after handling cats, especially before you handle or eat food. And especially, it's very important to do these preventive measures for pregnant women. Next one, Anisakis species are nematodes or roundworms that have been associated with foodborne infection. They generally cause foodborne disease through consumption of raw or undercooked seafood. The most common foods include seafood such as cod, haddock, fluke, salmon, herring, flounder, and monkfish. And in this picture, you can see transmission. This parasite is transferred in the water in which the marine animal lives. This parasite may also be transferred to other foods by improper food handling. And it's better to prevent, handle and cook seafood properly. Go on. Trichinella spiralis is a foodborne roundworm that can cause parasitic infection. People get trichinellosis by consuming raw or undercooked meats such as pork, bear, cougar, fox, wolf, or dog infected with Trichinella larvae. This parasite is inherently carried by animals. It is generally thought that a possible route to the animal is through consumption from eating infected tissues from other animals. You should know how to prevent is to cook pork until there are no signs of pink and always cook meats properly. Taenia cyaginata or solium. Humans are the definite hosts of both organisms. This means that the reproductive cycle and thus egg production by the organisms occurs only within humans. Eggs are passed in human feces and they may be shed into the environment for as long as the worms remain in the intestines, for as long as 30 years. In addition, the eggs may remain viable in the environment for many months. People get teniasis by consuming raw or undercooked infected beef or pork. Beef tapeworm Teniasis cyaginata infections appear within 10 to 14 weeks. Pork tapeworm infections appear within 8 to 12 weeks. How to prevent is to cook all raw beef and pork steaks, chops, and roasts to a minimum internal temperature of 63 degrees. Staphylococcus aureus. This is a type of foodborne intoxication is caused by consumption of food contaminated with staphylococcal enterotoxins produced by certain strains of Staphylococcus aureus while growing in food. The organism produces the following 5 serologically different enterotoxins that are involved in food-borne intoxication. Staphylococcal enterotoxin A, B, C, D, and E. Individual strains of Staphylococcus aureus may produce one or more of enterotoxin types while growing in food. Toxin production occurs at gross temperature 1244 degree, pH 4.2 and salt concentration of less than 10%. No toxic production occurs at temperatures below 12 degrees, pH less than 2.2, 4.2, and more than 10% salt. Flaky foods. Milk and milk products, including pasteurized milk, yogurt, chocolate milk, fermented milk, cream-filled pastries, poultry, fish, shellfish, egg production, etc., have been involved. Incubation period is 1-6 hours after consumption of food contamination, with at least 1 microgram of enterotoxin. In clinical signs include salivation, nausea, vomiting, abdominal cramps, sometimes diarrhea. Duration of illness is 24-72 hours. Some individuals may not always demonstrate all the symptoms associated with the illness. In more severe cases, headache, muscle cramping, and transient changes in blood pressure and pulse rate may occur. Recovery generally takes 2 days. However, it is not unusual to complete recovery to take 3 days and sometimes longer in severe cases. Bacillus cereus foodborne intoxication. The organism produces the following enterotoxins, which are involved in a foodborne intoxication. 2 diarrheal enterotoxins. Hemolysin enterotoxin, non-hemolytic enterotoxin, emetic toxin. Bacillus cereus is a common soil saprophyte and is easily spread to many types of foods, especially of plant origin. It's frequently isolated from meat, eggs, and dairy products. Cereal dishes like rice, spice, mashed potatoes, herbs, vegetables, minced meat, cream, and milk pudding have been involved in Bacillus cereus poisoning. The symptoms of Bacillus cereus diarrheal type food poisoning mimic those of Clostridium. The onset of watery diarrhea, abdominal cramps, and pain occurs 6-15 hours after consumption of contaminated food. Nausea may accompany diarrhea, but vomiting, emesis, rarely occurs. Symptoms persist for 24 hours in most instances. The syndrome is characterized by nausea, vomiting, abdominal cramps that occur 1-6 hours after consumption of contaminated food. Diarrhea syndrome. Guys, you should know, but sometimes when I ask you about foodborne diseases, and some students tell me that diarrhea is a foodborne disease. No, it's just syndrome. Please be careful, pay attention, and learn it, that diarrhea is a syndrome, it's not a disease. In the diarrhea syndrome, patients experience profuse diarrhea, watery stool, abdominal cramps, and tenesmus, rarely vomiting, beginning 8-16 hours after ingestion of contaminated food. Fever is absent and symptoms resolve within approximately 12 hours. You can see on the picture what's happened. Clostridium perfringens intoxication. Intoxication caused by Clostridium perfringens, enterotoxin produced in the gastrointestinal tract. The organism is found in the soil, dust, water, sewage, marine sediments, decaying materials, intestinal tracts of humans and other animals. If the spores are present as contaminants on raw meat, they may resist boiling or steaming for 4 or more hours, and on slow cooling, the spores will germinate into rapidly multiplying bacterial cells, which produce large amounts of toxin. The food involved are those that are prepared one day and served the next day. Foods that have been involved include red meats, chickens, fish, pork, fruits, vegetables, etc. Food poisoning occurs when the level reaches 178 cells per gram of food. Growth is enhanced by anaerobic conditions achieved after removal of oxygen by cooking. This illness is usually over within 24 hours, but less severe symptoms may persist in some individuals for 1 or 2 weeks. A few deaths can be reported as a result of dehydration and other complications. In most instances, the actual cause of poisoning is temperature abuse of prepared food. Small numbers of the organism are often present after cooking and multiply to food poisoning levels during cool down and storage of prepared foods. Symptoms: nausea, intestinal cramps, pronounced diarrhea, and vomiting is rare, and the illness takes a duration of 1 or 2 days. How to prevent: proper cooking of food and eating freshly prepared foods, fast cooling of cooked food. Storing food in small quantities will enhance cooling. Storage of leftovers or unused foods in freezers. Next one. Clostridium botulinum, foodborne intoxication. The strains are divided into proteolytic and non-proteolytic types according to whether they hydrolyze proteins or not. The intoxication is caused by botulinal toxins A, B, E, F, and G, produced by Clostridium botulinum type A, B, E, F, and G, while the organism grows in foods. Clostridium botulinum type C and D produce toxins C and D that cause disease in animals. Type E strains are not proteolytic, while the rest are proteolytic. Spores of Clostridium botulinum type A can survive temperatures of 120 degrees. Proteolytic strains grow at temperature range between 10 and 50 degrees, while non-proteolytic grow at 23-45, optimum 35-37. Toxin production occurs at temperature range between 25 and 30. Botulinum toxin is one of the most lethal pollutants known. The calculated lethal dose for an adult person is 10 micrograms. Foods associated with anaerobic conditions such as spoiled canned meat on hams and bacon stacked without air access are particularly liable to be infective. Homemade fermented foods have been incriminated together with smoked, pickled, and canned foods that are allowed to stand and then eaten without adequate cooking. Uncooked fish, fresh foods, and safe because they are eaten before the toxin has had time to develop. There are two different illnesses: adult botulism and infant botulism. An adult may become ill by eating spoiled food containing the botulism toxin. This toxin is produced when the bacteria grow in improperly canned foods and occasionally in contaminated fish. Symptoms: nausea, vomiting, fatigue, dizziness, etc. 1-10 days. Infant botulism is caused by eating the spores of the botulinum bacterium. For infants, one source of these spores is honey. When contaminated food is eaten by adults, toxin is absorbed from the intestines and attached to the nerves, causing the signs and symptoms of botulism. Early symptoms include blurred vision, dry mouth, difficulty in swallowing or speaking, general weakness, and shortness of breath. The illness may progress to complete paralysis, respiratory failure, and death. When infants eat contaminated food, the spores grow in the intestines and release toxin. Preventive measures are proper manufacturing practices, ensure proper sterilization and preservation of canned food. Preserved food processing, rancid, or other odors should be rejected. Fungal intoxications. They are caused by consumption of metabolites produced by fungi when growing in food. These metabolites are called mycotoxins or mycotoxins. Grains, oilseeds, fruits, and vegetables are mostly involved if they are stored at high humidity or if they are not properly dried before stage. Alpha-toxicosis is caused by alpha-toxins produced by the fungi Aspergillus flavus. There are 4 types. When consumed in large doses, they are lethal in causing acute hemorrhagic syndromes. Long-term consumption of small doses cause liver tumors, as these are potent cancerogens, as you can see on the picture. The sources. Prevention of alpha-toxicosis, proper drying and storage of grains and other affected foods, quality control of potentially hazardous foods, and use of fungicides as seed dressing to protect source cereals. Thanks for attention. If you have any question, you can ask your teacher or me, and see you all question and more information on our page on Instagram, Cathedral Hygiene on Meadow. Thank you very much.